This lecture is going to give you a brief overview of functional groups and their basic characteristics. So if we think about an organic molecule, organic molecules are all based on carbons. So we'll just think of it as a carbon chain. And if we make what we call a hydrocarbon, that's carbon with hydrogens on it. So this is kind of the basic structure of all the molecules of life, organic molecules. And in order to make different organic molecules, which have different characteristics, like carbohydrates, lipids, proteins, and nucleic acids, what ends up happening is that you take one of these hydrogens and you replace it with a different functional group. So that's going to be one of these 7 functional groups here. So let's say we've taken away that hydrogen and we're going to put a hydroxyl group on it. In that case, it's an OH group, which I can write like this, or I could have written like that. So it's an oxygen bound to the hydrogen. This is the OH. So that's a hydroxyl group. And so if we replace a number of these different hydrogens with hydroxyl groups, all of a sudden we'd have something we call an alcohol. If we were to take other hydrogens away and replace them with other types of groups, we get different types of molecules. So as we're going along and you're learning about the 4 large classes of molecules of living organisms, pay attention to which of these functional groups are present and which are not, which will help you identify them if you have to look at pictures. So here we have a nice chart. They're showing you the ball and stick model of that functional group, as well as the structural formula. And then they give you an example here, highlighting the functional group in that molecule. And then they tell you, if you're only taking into consideration this one functional group, what kind of compound would you have? Now, if you combine them, you start combining these in different ways, like let's say you have hydroxyl groups and carbonyls, now you're going to start getting different types of molecules on top of that. Like if you have hydroxyls and carbonyl groups, you're getting a carbohydrate. So what I have here for you is these electron density maps. So the way that this color scheme works is that the red areas are high electron density. So these are where the electrons are concentrated. And the areas that are blue are the low electron density. So that's the areas where electrons are lacking. And of course that's due to electronegativity. So anything with the red is going to be having an electronegative atom under there. So this is an oxygen, which makes sense because oxygen is much stronger than hydrogen or carbon with electronegativity, so it pulls the electrons onto itself. And the reason I go over this and show you these as we go is because electron distribution is the reason that a hydroxyl group behaves differently than a carboxyl group, that behaves differently than a carbonyl and an amino group. And depending on sometimes the state of the molecule, so in this case there's only 2 hydrogens here. Let me see if I can zoom in. There we go. So here is your oxygen, and here's your 2 hydrogens. And in that case, you can see the oxygen has the electron density for this amino group. But now you added another hydrogen, and here's your 3rd hydrogen on your oxygen, and now all of a sudden the electron density is no longer on that region. And here are some of the last groups. These are all in your book, so you should take a look at them. So let's go through one by one. So hydroxyl, hydrogen, and oxygen. Hydrogen, oxygen is how you can remember that one. So that's what it's made of. Sometimes we write it like this without the line in between the oxygen and the hydrogen, but in fact there is a bond there. And if you only have hydroxyl groups in your molecule, you're going to have an alcohol. Now, hydroxyl groups are also present in other molecules like carbohydrates, but there'll all be additional groups as well. So this is an example of methanol, which is it has a hydroxyl group on it. This one's an alcohol, and you can see that this is polar, right? So we have a high electron density and a low electron density, red and a blue. And so what ends up happening is a hydroxyl group is a polar group, because the oxygen is pulling the electron density off the atoms around it. So this means they're going to dissolve well in water because it can form hydrogen bonds, because it's polar. Carbonyl groups, and that's how you say this, carbonyl, is a carbon double bonded to an oxygen. Carbonyl groups can be either on the end of a molecule or in the middle of a molecule. So both of these are types of carbonyls. Specifically, the ones in the middle are called ketones, like a lock and key, where your key goes in the middle of the lock. And the ones on the end are called aldehydes. These are also polar. You can see that the oxygen is still more electronegative than the carbon, and so it still has this unequal distribution of electrons, which also means these are going to be pretty dissolvable in water. Carboxyl groups look like a hydroxyl and a carbonyl, but it doesn't count as either of them. When you put everything together, you get a very different characteristic than you did before. So we can actually think of this as the choo-choo group. So CHOO, CHOO. And I remember that just because a boxcar is part of a train. So the carboxyl group is the choo-choo group, the train group. So in this case, what we have here is some extreme electron distribution differences. So you can see in this case, we have two oxygens. Both of them are pulling electron density onto themselves. This hydrogen, which is like this one here, has very little electron density, so it's not held on very tightly at all. And what happens actually is that you can easily lose that hydrogen to the surrounding solution. And remember, if you lose a hydrogen, that's increasing the amount of hydrogen ions floating around in the solution, which makes it more acidic. So it increases and makes the solution more acidic, so it is called an acid. So then they become ions. As you can see, there's a negative charge here. And they'd go back and forth between these two forms. An amino group is the nitrogen and two hydrogens. And we're going to see that these are particularly important in proteins, in amino acids, hence the name amino acid. The amino group is going to start out with most of its electron density concentrated on the nitrogen group here, pulling it away from the hydrogens. Nitrogen's also very electronegative. So what you're going to actually be able to see is that the nitrogen can pick up a hydrogen ion. The red spot here is the nitrogen is very strong. It attracts hydrogen ions very strongly, and so it can actually bind those and ends up getting an additional positive charge. So now this is positively charged. So it's sucked up the hydrogens from the solution. We call that a base. And then it goes back and forth. So then we have sulfhydryl groups, which are made of sulfur and hydrogen, sulfhydryl. And these are going to be really important for building proteins. So the most important protein building block that has this is called cysteine. So cysteine is what we call an amino acid. Interestingly enough, if you can look here, cysteine has a number of functional groups, right? It has its little carbons in its backbone, but really then one functional group over here, this is a choo-choo group, so that's your carboxyl. Another functional group over here, it's your nitrogen with two hydrogens, that's an amino group. And then in this case, the final group that we have here, there we go, is our sulfhydryl group. So the sulfur is going to be able to bind with other sulfurs to hold the shape of a protein together. So let's say this is our protein, and we want it to hold together in this kind of looping structure. If we have a cysteine on one side over here, let's say this is its group, this sulfhydryl group, and we have a cysteine over here that its sulfhydryl group is like that. What can actually happen is that a reaction is catalyzed and a bond is made between the two sulfurs, and now you have this covalent bond helping hold the protein in this bent shape. So when we say disulfide bridge, di meaning two sulfurs, disulfide bridge, this is what we're talking about. And so that's going to help hold the protein shape together. Now, most of the protein is held together by ionic and van der Waals forces, hydrophobic interactions, polar dipole-dipole interactions, hydrogen bonds. But these are also very important. So that's why we care about sulfhydryl groups. So then we have phosphate groups. You can see they're pretty large. They have a phosphorus in the middle and 4 oxygens surrounding them. In some conditions, phosphates can have the hydrogens attached to their oxygens. But typically in biological systems, what we see is that the hydrogens have fallen off, and so we get negative charges. And so those negative charges are going to account for properties of DNA, because phosphates are in nucleic acids, and DNA and RNA and ATP. And so we're going to see that to be a big role there. Then finally we have methyl groups. So methyl is like methane, CH4, but methyl is attached to something else. So it just has the carbon and then the 3 hydrogens, and then it's attached to another entity over here. So here's an example of a methyl group. Methyl groups are found on a number of molecules, but a particular function that they perform is affecting this expression of genes. So actually, if you have DNA, in order to regulate gene expression, you can do a number of different things, but one of the things that we've actually found that can happen is the DNA can get methyl groups put on it. Call that methylating the DNA. And this helps to regulate whether the genes are turned on or off. You can also methylate the parts of the proteins that help curl DNA up. So DNA is curled up in the nucleus, and you can put methyl groups on that, those proteins as well, to help it curl or uncurl. So those are our 7 groups, and I'll just show you this series of pictures to show you why functional groups are so important. So we have a steroid here because it has these 4 fused rings. That's the characteristic of a steroid, a lipid. And you can see we have some functional groups here. So we have an OH, a hydroxyl group. We have a CH3, that's a methyl group. And we have another hydroxyl group in this backbone. Here, instead of a hydroxyl group, we have a carbonyl. So it's a carbon, remember at the corners are carbons, attached double bonded to an oxygen, so that's a carbonyl. We again have a methyl group and a hydroxyl group. Actually, look, we have additionally an extra methyl group here, where that's just hydrogen right there. That's what that would, it doesn't say it, but that's just hydrogen. And the difference between these 2 molecules is pretty great in effect. So estradiol, which is a form of estrogen, that's the female secondary sex characteristic hormone, and testosterone is the male secondary sex characteristic hormone. And so you can see actually the shapes are pretty similar, but the fact of this extra methyl group and missing methyl group here, and then a hydroxyl versus the carbonyl, makes them extremely different in function, because shape-wise, this shape turns out to be quite different than this shape as far as what can interact with what other molecules. So those are your 7 functional groups. So what you need to know is the names of those functional groups. It'd be good if you knew how to actually pronounce them as well. You need to know what they look like in a molecule and on their own. You need to know what their basic characteristics are. Are they polar and going to hydrogen bond and mix well with water? Are they nonpolar, like your methyl group, and they're not going to mix well with water? Are they going to be acidic, like your carboxyl group, or basic, like your amino group, that kind of thing? And then you'd want to know, when you look at your 4 large molecules of life, you're going to want to associate functional groups with those different molecules. For example, if you're looking for carbohydrates, you'd want to know that you're looking for hydroxyl groups and carbonyl groups. That's going to tell you, oh, that's a carbohydrate. If you're looking for a protein, PTN is protein, you're going to be looking for amino groups and our choo-choo carboxyl groups. There could be other groups in there as well, but those 2 are definitely going to tell you there's proteins involved. If you're going to look for a nucleic acid, like DNA or RNA or ATP, you're going to be looking for phosphate groups. Phosphate groups may also be on some types of lipids. So lipids are going to have lots of methyl groups because they're nonpolar. So the methyl groups, and sometimes if you have the phosphate, it'll be a phospholipid, so you'll learn about those types of things. So basically, you'll want to be able to recognize functional groups, identify a functional group, and realize what large molecule that means the picture is, and understand what characteristics that molecule is going to have, depending on which of these groups is present.